Okay, we might get started. Thanks everyone for joining. Again, this is another session of our Spatial Conversations webinar series. I'm Brett, the Market Development Manager for ANZ for Spatial, and my colleague.
Hi, I'm Kristen. I'm the channel business executive for New South Wales, Queensland, and ACT in Australia.
And for those of you who are new to the series, the idea is to get more background information, unpublished details about performing spatial experiments sort of into the public domain. We hope you find the series valuable, and we encourage you to ask as many questions of our speakers as possible. I know we have a very targeted talk today, so hopefully you'll be able to find out something very interesting for your particular field. Obviously, our speakers have spent a lot of time and have a lot of learnings to share. So we really encourage you to ask questions and maybe hopefully come off mute and ask them at the end of the call.
So with that, we would like to introduce our speaker, Lipin Loo, who will be talking on mapping neuropathic pain with integrated transcriptomics. Lipin is the group leader of the RNA therapies group and also a co-leader of the single cell and spatial biology node at the CPC in the University of Sydney. So many of you may already know him. He utilizes genetic screens and single cell and spatial transcriptomics to identify and map transcriptational changes underlying pathophysiological conditions such as chronic pain and neuropathy, elucidating these orchestrated transcriptional. I'm just not going to say that word for the rest of the, the webinar. Changes will have a far-ranging impact on our understanding of disease causing aberrations and instruct our strategies in restoring normal function in disease cells. So with that, as Brett said, at the Q&A at the end of the session, please feel free to put your questions into the chat, but also raise your hand and we can unmute you to ask your questions yourself. So thank you, Lipin.
Yeah, thanks. I'm gonna change my biography next time to have less words that are hard to pronounce. My bad. Thanks for the
You're free
introduction.
today.
Thanks for inviting me. I also did change like my title a little bit, just because I think for the crowd today, for the attendees, it might not make that much sense to go into chronic pain and neuropathic pain and all of that. So I changed it to uncovering molecular signatures of neurological disorders through spatial transcriptomics, because this is a spatial talk. And this is like an illustration I made from DALI, the AI platform, that basically is an impression of what Surat would think of like discovery at the lab bench, and try to do it a bit like for a spatial, like, you're sectioning tissue. In the past, you just look under a microscope with a H&E stain, but now, we can look at, specific cell types through transcriptomics. And I think, that's historically how far we've advanced is quite amazing. So I did say I didn't want to spend too much time on the science, but just wanted to talk a little bit about my journey from single cell to spatial. I'm interested in chronic pain. I'm interested in a specific form of chronic pain, which is neuropathic pain. I'm interested in looking at changes that are happening in the spinal cord of neuropathic pain models. And so I started out doing single cell. This was single nucleus data that we generated using 10x genomics, a 3' capture kit. From that, we're able to identify gene expression changes in specific clusters. And then we were interested in finding out, like, okay, what are these neurons that are affected in neuropathic pain? And so the next thing we did was spatial transcriptomics, and this was done using the original Visium method, where I believe each probe like area is probably about like 100 microns. I might be wrong on that, but it's like it's not true single cell. Yeah, sorry.
50 microns. Yeah.
Yeah, 50 microns. Yeah. But then the space on the side as well, I think it's about 100, 120. So it's not true single cell resolution, but we were able to use this to identify our neurons in the spinal cord, like figuring out where they sit within the spinal cord, and then mapping out like how they interplay and stuff. So this paper is under review right now. But while we were doing this, we noticed we needed something that's higher resolution. We needed to look into those specific neurons in the spinal cord and probe for these specific gene expression changes that we've observed in single cell. And so to do that, we use Xenium. If I'm just going to give a brief summary of this. Maybe a lot of you probably already know how Xenium works, but if you don't, this is a brief summary which I've taken from the 10x website. You basically have your fixed tissue, whether it's FF or FFPE. You section them and you can place them within the capture area. And then you can then undergo deparaffinization or decross-linking if it's FFPE. That's what we did. And then there's a workflow for probe hybridization and then a ligation. So the way the Xenium workflow works is you don't just have one probe like your normal in situ hybridization. For higher fidelity or accuracy in transcript calling, you have multiple probes. In this case, 2 probes for each transcript. And then only when both probes are present, you can do the ligation. And then once it's ligated, you can do the rolling circle amplification, basically generating a lot more material from that. And then this is like an overnight process. The next morning, you take your slides, you put it on the Xenium analyzer, and then you incubate with fluorescent probes. These fluorescent probes will then bind to your product. And then you can then cycle through, well, you don't do anything. You actually just hit start run on the Xenium analyzer, and then it does everything for you. And then they can image, and then kind of like similar like NGS, like next gen sequencing, where they can now call basically like if you have these certain combinations of fluorescent probes, they can call it back to a certain transcript that you've detected, and then you can visualize this using like a software. Before I jump into the data, I also need to mention a little bit, because I think this is more about this session is more about talking like the workflow, how do you actually design your experiments. So for the Xenium, you can either use an available panel that you can just buy from 10x, or if you, like me, I'm interested in very specific genes and specific neuronal clusters in the spinal cord, I had to make my own panel. And it's quite easy. You just go 10x Genomics Cloud. They have a website now. You create an account. Just showing a screenshot here that I pulled from the cloud. You go into the panel designer, and some requirements for making your own panel includes having a reference data set. So in my case, because I had my single nucleus RNA-seq data, I would use that and the cell annotation attached to that data set to develop or build my own panel. And then there's a bioinformatics expert from 10x Genomics that will reach out and then confirm whether your distribution of your probes that you've designed makes sense, because sometimes you don't want something that's too abundant or too rare, because then you got to adjust the number of probes based on their abundance, basically. You don't have to do this. The bioinformatics person from 10x will actually do that for you, but you do need to have a reference. If you don't have one generated in your lab, you can also use something that's publicly available. If it's published, you can just like download that. So we did that. And this is like an example of like a spinal cord that we did with Xenium. You can see all these different transcripts and they map to the areas that you would expect them to. This is the dorsal horn, the ventral horn of the spinal cord. We've got glutamatergic markers, GABAergic markers, motor neurons, the cholinergic markers, astrocytes, microglia, myelin basic protein. This is the ependymal cell marker. And then here at the bottom here from MAF onwards, NTS, NMU, TAC2, TAC1, these are layer specific markers. And we can see quite nicely like the distribution of that. And then these are some rarer transcripts that we weren't able to see previously with just Visium. But using this high resolution probe method, we're now able to see these transcripts with Xenium. And I was also asked to share like how I did this. Basically, I used the Seurat package, which you can just go to this link down here and just follow the vignette, and you should be able to load your Xenium file and be able to follow the instructions and visualize your transcripts. What's more interesting though is after you've got all these transcripts, you can still do some machine learning annotation. And in this case, I did deconvolution with RCTD, which you can find in this GitHub link here. So what it does is it takes my single nucleus RNA-seq data and then labels our xenium data. And here I have two spinal cords. One is the sham control healthy spinal cord, and then I have the spared nerve injury spinal cord where these mice have neuropathic pain. And one thing that we noticed immediately was that we're able to, because of the way I developed this panel, I'm supposed to be able to see these important neurons that are affected. But more importantly, we can look and see these transcripts of interest are highly elevated in the neuropathic spinal cord. And then going one step further, before I jump to that, while playing around on the 10x Genomics cloud, I noticed that 10x is now providing cell type annotation. So maybe in the near future, you might not even need to run your own deconvolution with RCTD. You might be able to do it with the directly on 10x Genomics cloud. So going one step further, because we're able to identify our cell types, we can ask specifically within those cell types, can we compare the transcripts and then do some stats? And that's what we did here. We look at different clusters of cells, glutamatergic cluster 6, GABAergic cluster 4, and microglia cells, and we could see our genes of interest being elevated in these sections. And then I was also asked to touch a little bit on Visium HD before I wrap up. I have not done anything spinal cord wise on the Visium HD, but we're also developing some new therapies for pain and looking for changes in the brain. So I have done some Visium HD on brain tissue, which I'll share with you. So the difference between Visium HD and Xenium is with Xenium, it's an imaging based method, whereas Visium is actually sequencing based. So you first prepare your sections on just regular slides. Unlike in the past, if you use the first generation Visium, you need to place your tissue directly in the capture area where it has these probes on the slide, on the Visium slide. And in the past, I've had some issues with spinal cord like floating off in the process of one of the steps between like 3 cross-linking or like from the probe hybridization workflow that deemed it like unfeasible. I wasn't able to basically do that experiment because once your tissue floats off, you get like dispersal of like your transcripts. And so it's not really that good when you're trying to then visualize your image. But using now CytAssist, you can just put your tissue on a regular SuperFrost slide, and then you can do most of the workflow, like the staining, and then I think even the probe hybridization can be done on that prior to transfer. I could be wrong on that.
That's right.
Yeah, that's right. Yeah, and then once you've done all of that, you can then sandwich your SuperFrost slides with the capture slide on the CytAssist. And then once you've done that, I think I forgot how long was the workflow, but basically it then releases, once you've sandwiched it, there's a release of your transcripts onto the capture area, which is on the HD slide. Here you have a lawn of like these probes that have the UMI spatial barcode and oligo DT that captures your polyadenylated transcripts. And then, once you get that, it's really just the workflow is the same as preparing your library from the single nucleus RNA-seq protocol. You prepare a library, you introduce index sequences, and then you sequence it with NGS. And then when you get it back, in the past, we would have to go onto our HPC at the university and then like do alignment, and then like after alignment, you pull that off and then you do it on your computer, like you do the downstream workflow. But what's cool now is that on 10x Genomics Cloud, you don't have to, try to engage the university to get access to the HPC. You can directly just upload your files to the 10x Genomics Cloud. And this is what we did here in for our brain study. After sequencing, I just uploaded it onto the cloud, and then I think this only took like a couple of hours, and then alignment was complete. You get your QC report, like your web summary file, as you would normally get in your single cell experiments, but here you get it for your spatial experiments, and then you would get something like this, like a web summary file. So the metrics here, so shown here is a brain section that we did. It was just like a sagittal section, not the normal coronal that you guys are probably more accustomed to seeing. But you get a bunch of metrics, like how many number of like 8-micron bin squares are detected under the tissue, how many UMIs are detected per bin, how many reads from the sequencing, and how many total genes were detected. You can see like it actually did pretty well, like 99.1% of our reads were mapped to the probe set, and then they were mapped confidently. And then, yeah, these are the total number of reads that we sequenced for this experiment. You can find this recommendation on the 10x site, like depending on the area of coverage of your tissue, you can calculate how many reads you should sequence to give you optimal results. So after you get this data, you can then pull it out. Like there's a loop, C loop file. So this is an 8-micron bin file that you can then put on the loop browser. And then 10x, like during the alignment, it also does clustering for you. And then basically it showed me that I had like, depending on like the graph-based method, this gave me about 15 clusters. And then there's also a bunch of stuff that you can do in there, looking at differential expression output. But I personally like doing this on Surat, and so I've taken that file and then loaded it into Surat, and then unfortunately this is as far as I've gone on that. I haven't done more, but basically you can see this is a GABAergic marker that depending on how you bin it, like 16 or 8 microns, you can see the expression within the tissue. So yeah, I think I only have 15 minutes. I went slightly over, but that's my presentation, and I'll open it up to questions from the audience. Thank you.
Thanks very much for that, Lipin.
Yeah, no worries.
If anyone would like to ask a question, please feel free to come off mute or to put your question into the chat. I'm just looking at it now. I don't see any at the moment. So I might ask one question, Lipin. I mean, you talked about getting spinal cord onto the slides. Most people think of it as a difficult tissue. For Xenium, how did you find getting the tissue onto the slides?
So previously, I guess it depends on what tissue you're working with, like human or mouse. We had issues when it was with the human tissue. I think it's because it was much bigger than the mouse tissue, and as a result, the spinal cord's highly myelinated. There's a lot of fat content within that tissue. And as a result, like when we were working with the human tissue, it did tend to float off when we were doing the original Visium protocol. But having said that, with the new side assist, everything's done on the regular Superfrost slides. We haven't had any issues with like either human or mouse tissue, just because I think the electrostatic charges are much stronger when you're just using a regular slide, whereas previously if you're trying to put it directly on a capture area that has probes, I think there was probably slight less attraction between your tissue and the capture slide, and as a result, we were seeing a lot of sections floating off. But using this new workflow with side assist, I don't think we've had any issues of sections floating off at all.
Okay. And for Xenium, did you find, I mean, the mouse work you did with Xenium, how did that go?
Oh, I mean, Xenium doesn't. I think a lot of the floating off issue is the repulsion of the probe on the slide, and then with Xenium, you don't. There aren't actually any probes on the slides, right? It's just really a slide that allows you to attach your tissue to, and then the probes come in on top of that for the imaging. So we didn't have any issues either with the Xenium. In fact, like we were able to place okay, because we're trying to maximize the output that we get from each capture area. we placed like almost 15 mouse spinal cord sections in each capture area. So we got like ton of data out from the Xenium, and none of
Right.
those sections came off during the imaging. Yeah, so Xenium was like smooth sailing. Like we got data out from the first run, and then we just did a couple more runs after that, and everything was fine.
Fantastic. I know we have a
Yeah.
question from
Yeah.
Klaus who would like to talk. I'll just unmute him.
There we go.
Hello, does it work?
Yes,
Yep.
we can hear
Yep.
you, Klaus.
Can we hear you?
Fantastic talk, Lipin.
Yep.
Sorry, I'm Klaus Lehner from the University of Auckland in New Zealand.
Oh, nice to meet you.
Fantastic talk. Looks like you're in a very similar spot in your 10x journey compared to us, or as us. I have a very peculiar question to your slide 8, if I counted right.
Okay, let me
which
see
I think
slide
was
8.
your first 10x HD result slide.
Oh yeah, yep.
It's a very peculiar question. I apologize in advance.
Uh-huh.
It says on the run title, olfactory bulb, Visium HD
Yeah.
on control. Are you
Yeah.
working with olfactory bulb
Yes, I am.
in human?
No, this is mouse. We're only working in mouse right now. We're developing a therapy, which I unfortunately can't disclose. But we think it's targeted to the olfactory bulb, and so we're using spatial to see if we could detect our therapy or changes that are happening in olfactory bulb.
Okay, that.
Yeah.
I wonder whether the 10x people can exchange our contact details if you
Oh
can.
yeah, sure.
I'd really love to talk to you about olfactory bulb.
Yeah.
We're working with human exclusively.
Okay.
So that would be fantastic. And I
Yeah,
don't
it could
mind
be
talking
some.
about it here, but I think it interests nobody else but the two of us.
Yeah,
Karen,
it is a very interesting area. Like we only got into this because like a postdoc noticed that we were getting presence of a therapy that we're trying to introduce in the olfactory bulb, and so we started investigating deeper into that. We didn't start out like looking at the olfactory bulb, but because we're seeing presence there, we think it might be something worth pursuing. Yeah.
Well, wonderful. Yeah.
Yeah.
Karen, if you don't mind exchanging the contact details between the two
Yeah,
Yeah,
of us.
we'll
we can
organize that
definitely do
for you,
that.
Klaus.
Thank you.
Yeah. Thanks, Klaus.
Not a
Yep.
problem. We have another question just asking about the roadmap for Visium HD. At the moment, the only guidance that we're giving for Visium HD is that we are coming out with a Poly A capture version of Visium HD. So you'll be able to go beyond just the mouse and human probe sets that are currently available. So we don't have any guidance on a larger capture area, though we do understand that there's a lot of people interested and that the capture area is on a lot of people's minds. So hopefully we'll have some more updates in the future in that area.
And then, I mean, just can I just add
Yeah.
something real quick? So people always ask me like, why do you do Visium HD versus Xenium? And you know, because Xenium now also has like a 5,000 gene panel, right?
Yeah.
Yeah. What do you have an answer for that? Like, why should I do Xenium versus Visium HD?
I mean, there are a couple of big things that you pointed out. Obviously, Xenium has an analyzable area of about 2.3 centimeters squared versus the 6.5 by 6.5 millimeters that you get with Visium HD. So it has a significantly larger area for either a larger tissue or for more tissues. I think the data analysis is a little simpler with Xenium. So segmentation is certainly more advanced with Xenium than it is with Visium HD. Obviously, HD is new, and it is a grid-based detection format. So, you know, we have recommendations on how to approach that, but I think they're more advanced with Xenium. And we find with Xenium, it's a little bit more sensitive than Visium HD. I think sort of in line with what you saw in your own work, that Xenium, you were able to see things a little bit better. It's not a huge gap, but it is there.
Yep.
I think
I
Klaus
think
was
Klaus is asking another question.
yeah.
If I can, I don't know how many people I'm blocking with me asking questions. Lipin, I wonder, can you comment? Sorry, in the first instance, did you use what's it called, a multimodal stain for your Xenium work?
No, so I was quite early.
Okay.
So all we had was the nuclei stain. So the segmentation that we did was primarily using the expansion from the nuclear. Yeah, but we had a spatial meeting earlier this year with like other users, and I think they were forgot who it was, but someone in Melbourne did a benchmarking of like the different stains or like different methods of segmentation. And they said that the nuclei expansion method actually works as well as like the multimodal stain. Just
All right.
because if you're very conservative in what you're calling, like, I don't know, 5 microns away from your nuclei is a cell, or our cells, I think you run a lower risk of like having doublets or something segmented together.
For neuronal tissues where we might actually not the nuclei have in the section at all, this multimodal stain could be quite useful. But its overall performance for nuclear tissue doesn't seem to be quite as high as I think it was liver or the non-nuclear
Yeah,
tissues.
that's true. You're right. I don't remember what tissue they were looking at. But yeah, unfortunately, like with this, we were quite early, and so we only had the nuclei stain. But I think moving forward, we definitely use the multimodal staining. And then yeah, I think the segmentation might be better there for neuronal tissue. Yeah.
Do you have immediate plans for another Xenium run?
Yeah, well, I'm trying to just get this paper published. And right now, no. But we do have follow-ups, like, after wrapping up this paper, maybe next year we might do something. But yeah, for now, just trying to wrap up the paper.
Well, we were lucky
Yeah.
enough to win one of the Millennium subsidized runs at SHEC. So that's
Oh, yeah.
hopefully happen with an olfactory tissue. So hopefully that's happening
Okay.
before the end of the year. So
Yeah, that's
we
great.
might learn something
Yeah.
with the multimodal stain. Cool. Thanks, Millennium, again.
Thank you, Klaus. We're almost at time. I'm just going to ask one question because there's always comes up, and I know you've sort of alluded to it in your talk. Many people are always asking about software analysis. Like, you were obviously fairly experienced with single cell analysis. How did you find transitioning your experience from single cell to spatial using Seurat?
If you know how to run Seurat for single cell, the jump is not that, like, I mean, that learning curve is not as steep. It's basically just following. But if you don't know how to run anything on R, it can be quite difficult. Like, you need to learn how to operate that. But if you have that basic skills of using R and plotting things, it's quite easy. You just load your libraries and follow the instructions. But having said that, there are a few things that will pop up from time to time. If you're trying to deviate a little bit from like what Seurat is meant to do, you might need to write some codes and subset certain things a certain way for you to do the analysis. But yeah, like if you have some basic experience, quite easy.
Cool.
Yep.
All right, well, I think we're at time when we don't have any more questions.
Klaus has raised
but-
his hand, actually.
Klaus
So
has another one. We'll
Yeah,
take the last
Klaus.
Klaus question.
Not really a question for you, Lipin,
Yeah.
but maybe for our friends at 10x. The group around Fabian Theis in Munich have about a month or so ago published a rather impressive paper about a general framework for spatial data. Now they're obviously all keen on Python and the like. I wonder whether there are plans in 10x to support this as a data storage and analysis framework through things like API to raw 10x data. Have you heard of anything about this, David?
Yeah, I'm not aware. I know we are generally engaging with the community about how to store and manage these large data sets.
Yes.
Obviously, one of the advantages with 10x is our data sets are relatively compact compared to some of the other spatial data sets. But it is something we're talking to people about, and we're open to conversations, but I haven't got any specifics about, coming out with a recommendation just yet.
Okay. Well, that's a framework that includes things beyond the single section, like multi-section registrations and all these very complicated things. And it isn't a tribal proposal. I think it's just a generalized proposal, open to all kinds of technologies. So the reason I mention it, just in case nobody at TenX has noticed it, there might not be a need to develop a I don't know, boutique solution. I haven't used it myself at all, so I can't talk about its pros and cons. But it is a high quality proposal out there.
Well,
Yeah, I think I mentioned this before. It'd be great if like a lot of these downstream analysis can be built into the 10x Genomics cloud. So, you finish your alignment and then you just click, I want to analyze with the Surat, and then Surat, load the package, runs everything on the cloud and gives us. I mean, there are a few things that we would need to adjust for, like if you want certain plots for features or, clusters and stuff, but it'd be great if, we get all of that like built into it. And I don't see why that would be too hard, because a lot of the pipeline already does clustering, does k-means or graph-based clustering, and just incorporating some of Surat's workflow into it should be quite easy.
Yeah, I can say that we do get this question a lot too, so it's a good one just to quickly touch base on. Our informatics group, their sort of position is that they sort of feel that the algorithms around spatial analysis are changing and developing quite rapidly at the moment.
That is true.
And the feeling is that at this point in time, we don't feel confident that there's necessarily a best in class. And so we're sort of recommending that people look at the different algorithms and what's appropriate for their own work as a third-party tool. And we're not planning to develop those in-house at the moment. But of course, it's changing rapidly. That could change as well.
Mm-hmm.
You'd probably need a much bigger bioinformatics team than you currently have.
Yeah, I know they're one of the busiest groups in the company, as they are everywhere. So we certainly feel that as well.
Also, as a community, I think what the community needs is a sort of a flexible framework standard that can include other technologies as well.
Mm-hmm.
And that would be, I think, very difficult to be developed out of a single vendor, if I can call it that. Because the competition always finds out last what the new developments are.
Yeah, and we certainly feel that the community's probably going to take the lead on
yes,
making
which
some
is
of these.
I think
so
what
we're
they try to
certainly
do
open to that.
With this paper from Munich. Cool. Anyway, I stopped ranting now. Sorry, David.
Not a problem, Klaus. Thanks very much for your engagement. All right, everyone. Thank you very much for your time. We've gone a couple of minutes over, but I think they hopefully were interesting conversations. And yeah, we look forward to seeing you at our next webinar. Thanks very much, everyone. Bye.
Thank you. Bye.